Hello, welcome. I hope everyone's doing okay. Um, Today we're going to talk about animals. There will be specific instructions online for what your expectations are for the week. Think of the course as moving more towards a weekly type of system. So, anyways, we're going to start off with the basics of animals this week. Most animals are mobile and use traits such as strength, speed, toxins, or camouflage to detect, capture, and eat other organisms. And this has to go back to the idea that it's a kingdom of consumers. You could argue that, yes, fungi are consumers as well, but they absorb stuff kind of stationarily. They don't seek out and find things. So, what makes an animal an animal? Well, really, it has to move around and consume, and all of its adaptations are towards that end. So, for example, a chameleon captures insect prey with its long, sticky, quick moving tongue. Again, so what are the adaptations you think of when you think of a chameleon? Probably one of them is that it blends in. It tries to avoid being eaten by other animals, but also it is trying to avoid being detected. And so that's kind of both ends of the camouflage there. uh, Long, sticky, quick moving tongue. Again, the tongue wasn't long, it wouldn't have as much of a reach. The stickiness increases its chance of um, success, and it has a quick moving tongue. Again, speed is an important issue there. So there you go, picture of a chameleon. So here we look at the basic outline of animal development. This is going to be kind of where we talk about, what makes an animal an animal. Notice the organisms that we have here. Um, so I'm trying to get a pointer here, and it's not doing what I want it to do. Okay, let me end the show there real quick. Also, let me get rid of this beeping. Okay, now let me go back to this, and well, here you can see the pointer that I have going on here. So right here, notice that we include both sponges and jellyfish, and there's quite a difference there already. Like right here, we're talking about the fact that there's tissues versus no tissues. Um, and then here we have bilateral symmetry versus radial symmetry. Again, a major difference in animal development. And so you'll see that there's kind of this progression of, what's the first novel thing that makes an animal an animal. So maybe pause it a second and just come up with a list of a few things that you think make animals animals. Okay, so when we look at the sponge, you got to remember that the sponge is considered an animal by at least most scientists. There's some debate about it. And so a major issue is that there are exceptions to nearly every single criteria for distinguishing animals from other life forms. So there's a lot of a lot of exceptions, a lot of flexibility that needs to be here. But however, if we take all these characteristics together sufficiently, that defines a group. In other words, it's going to have most of these things um, in the group of animals. So first and foremost, they have to be heterotrophs. Like that's pretty much so straight across the board ish. When you guys start reading your book about corals, you'll see that there are some corals and such that do get some of their nutrition from a symbiotic algae that lives inside of them. They have to be multicellular. Again, pretty general rule of thumb. There are some single-celled life stages, so again, most of them are multicellular throughout their life cycle. They lack cell walls. Again, cell walls, we learned, are made out of a couple different materials depending on which kingdom you're talking about. When we look at animals, they do not have cell walls. Now, one thing you need to be careful about is that we talk about cellulose being the cell walls of plants, right? Well, there are some animals that do produce cellulose, not many though. So therefore, you do, again, you can't say they don't make cellulose because there is that exception there. Their bodies are held together by structural proteins such as collagen. So again, a structural protein, that's a pretty general term. Like that's not that specific, but they have something that kind of holds them together. Nervous tissue and muscle tissue are unique defining characteristics of animals. Do all animals have muscle tissue and nervous tissue? And you might say, yeah, I mean, every single animal I can think of that comes to mind has nervous and muscular tissue. Remember, we have to include sponges. So again, there are some exceptions. Tissues are groups of similar cells that act as a functional unit. And again, that is something that's pretty loosey-goosey in terms. Generally speaking, most animals have tissues, but again, there's an exception right off the bat. So really, what makes an animal an animal? If I'm summarizing it down, they have to eat other things. They have to be multicellular. They have to move around. Most animals reproduce sexually. Again, they have at least available to them the option of a sexual reproductive stage. After sperm fertilizes an egg, the zygote undergoes rapid division called cleavage. And again, all animals go through this division called cleavage, but you might, again, start to look at animals and say, well, they all are doing the same thing here. By the time you're at 8 cells, there's already a difference in the different major groups of animals if you go back to that cladogram a few pages ago. So cleavage leads to formulation of a multicellular hollow blastula. So yes, that is true. We do have that. A solid ball of cells is called a morula. Then we get to this hollow ball of cells called a blastula. The blastula undergoes gastrulation, which now, so we have this hollow ball of cells, and it's going to fold in on itself, and there's going to be an inner and an outer layer. And the gastrula has different layers of embryonic tissues. Now, already, we have to remember that sponges aren't going to have this. So sponges, basically, they become a morula, and I can't remember if they actually make it to the blastula stage or not, but that's pretty much it. And then they kind of go on their merry way, and they live the life of a sea sponge. Whereas this gastrulation is going to occur, and every animal is more advanced than that. So, again, kind of leading to that whole idea of animal phylogeny having the branches that it does. So, all animals, sperm fertilizes egg. We get to an 8-cell stage. Now again, here we already have 2 major groups, and there's 2 major differences. You can see here how the cells line up nice and neatly. They don't in all the groups. We get a ball of cells, a hollow ball of cells, and that's it. Now we have our sea sponge. But everything else that's in the animal, this was important. One side grows faster, and because it's growing faster, it pushes in, and now you have 2 germ layers. So basically, every animal that's more advanced than a sponge has 2 germ layers. They have an inside and an outside. So just think about this. What is on the inside of all animals, and what's on the outside of all animals? So if you were thinking the insides are their guts, good. I mean, that's the digestive system. The outsides are their skin. Now, if you look at this hollow ball of cells, if this is an animal, just imagine this being an animal, what do we have here? What's this little opening? Well, again, back to up here where we were talking about like the cells differentiate. So in our lower animals, this is going to develop into a mouth. And that's it. That's it for the digestive system. I mean, if you think about your digestive system, if your food were to go in here, where's it going to go from there? Well, it has to go back out that same opening. So the mouth and the anus are the same thing in these early vertebrates. And even then, eventually, this will form a tube, and then we'll have what's known as a complete digestive system. So this is incomplete at this time. Depending on how advanced the animal is, the more advanced ones get a complete tube, and they get a complete digestive system, and this is the mouth down here, and then the anus develops later. However, if we rotate these cells around just a little bit and make them not line up, that's a different group of animals. Those groups of animals, the anus develops first and the mouth develops later. Anyone who's already taken my anatomy class knows that this is not exactly true. I mean, it's true, but the development is even more complex than what we're making it out to be here. So if you take anatomy in the future or have already taken it, just remember this is a little bit more complex process when we look at the placentals. Most animals have at least one larval stage, and if you think about it, humans aren't exactly larval, but we mature them until they get to being able to be born, and really we're not self-sufficient once we're born. So in a way, we kind of have a larval stage, but a lot of them do. Like if you think about like butterflies, they obviously have a larva. Even frogs, they have a larval-like stage in the tadpoles. So they're distinct from the adult. And if we think about how are human babies different from adults, I mean, if your head was 1/3 of your body size, how big would your head be? It'd be gigantic, right? Well, I mean, really, our children, that's when they're born, their head is 1/3 of their body size. So they go through quite a bit of changes. So pretty much every animal goes through serious changes as they develop. A juvenile resembles an adult, but it's not quite yet sexually mature. So think of your preteen years, like when you were around 8 to 10, starting to resemble adult, and then even more so when you get between like 10 and 13, and then in your adolescence, again, now you're becoming sexually mature and basically young adults. Most animals and only animals have Hox genes that regulate the development and form the body. So we see these Hox genes in animals, and basically this is going to direct our development. If you think of the ball of cells back here as like a globe, you're going to end up basically with a coordinate system where this is like your latitude and this is your longitude. And down here, all right, so let's just say we're developing an invertebrate. So we have the mouth here. How does it know to develop the mouth there? How does it know to start growing and folding in? And that has to do with those Hox genes. And different Hox genes start giving us different characters, different traits, and they kind of direct stuff to start growing at different times. So there has to be like this coordinate mapping that relies on being able to tell the cells down here, hey, you guys need to grow faster than these ones up here. So here we have the mouth, all right. Now let's just say we wanted an arm coming off this direction and an arm coming off this direction, there and there. So we're gonna have some type of quadruped with 4 arms, right? So how does it know to start growing these appendages? All right, and by the way, there's something seriously wrong with what I just said, because quadrupeds wouldn't grow this way, because they're going to be the group of animals that develop the anus first and then the mouth later. But just say, hey, we want to develop legs. Okay, that's really the point of where I'm going with this. So if we want a leg here, we got to tell these to start growing, and then tell these cells right here to start growing. And they've got to grow faster and stick out and then basically form limbs. And if we want more sections of this ball of cells to start growing limbs, you basically need to have more copies of this Hox gene directing more cells to grow faster and develop more limbs. And so these repetitions do things like direct how long they grow for what amount of time, at what stage they begin growing, and then where they're at in the coordinates of the cell or the cells on that blastula. So although the Hox family of genes has been highly conserved, in other words, basically an eye gene is an eye gene. You can actually take the eye genes off of a fruit fly, which has a very different eye from a mouse, and then take the eye gene of a mouse, remove the eye gene of the mouse, and insert the eye gene of the frog. And you might, remove the eye gene of the mouse and put in the eye gene of the fly. And you might think, well, is the mouse going to end up with fruit fly eyes? No, it ends up with mouse eyes. And it used the fly Hox gene that's in charge of eye development to make those eyes. So they're highly conserved. There's very few differences in the genetic code between the genes that make our eyes and the genes that make the eyes of a fly, even though the fly's eyes are very, very different. And early scientists really struggled with this because they thought, well, evolution was impossible because you'd have to re-evolve the eye several times because there's so many variations on eyes. And it turns out, no, really not. We just reuse the genes and just reuse them in slightly different ways. And that's kind of what's led to this simplicity of animals, is that we can use these same genes, the same basic set of genes. They call it the genetic toolkit. These toolkit genes can be used, again, just in different ways, and by having different numbers of repetitions and just very small variations. We're talking 1-letter difference. And it can basically give you a wide variety of things by just using a small set of genes, which are the Hox genes. More than 1.3 million animal species have been named to date. This is as of 2014. And far more are estimated to exist. Most scientists think at least 2 million, if not 3 million animals exist on the planet. And we might say, well, where are all these animals? Remember, you're thinking, like when I tell you to think of an animal, you probably are thinking of a horse or a cow, maybe your pet dog. But remember that vertebrates are one small group of animals, and they make up less than 5% of all animal species. The common ancestor of all animals likely lived between 700 and 770 million years ago. Um, and so remember, back to our analogy about the football field, we're talking about a very, very, very small amount of time. Within a few yards, if not within the 1 yard line of existence. If you take 770 million and divide by 4.5 billion, give you an idea of how long animals have been in existence on this planet. And we're talking, again, probably just a couple of inches, maybe a couple feet. So morphological and molecular evidence points to a group of protists called choanoflagellates. So these guys basically are really closely related genetically to our first group of animals that we're going to talk about, which are the sponges. In fact, a lot of scientists say, well, sponges really probably shouldn't be animals. Maybe they should just be a group of protists. But really, if you remember, we're just really advanced protists that have some novel adaptations. So the common ancestors may have resembled modern-day choanoflagellates. Choanoflagellates basically are these they basically are collared flagellates. So we have this flagellate, and they've got a collar, and they've got this flagella, and basically they trap food and they feed it. And we can see these same types of cells called choanocytes in sea sponges. Now, sea sponges also have a type of cell called an amoebocyte. So again, connections to protist backgrounds. And then we have all these other animals. So here you have sponges, and they have, basically they've got an inner cavity, right? They've got an ability to have an empty space. They've got different cells doing different things, but do they really have tissues? Think about it this way. If we take any of these animals down here and we put them in a blender, they're going to be done. Like they're dead. Sponges, any of these cells can become any other cell. Like you can blend up a sponge and it will grow into new sponges. You can't do that with these lower level invertebrates. I mean, these guys are really good at regenerating. We can cut this worm here in half and end up with 2 worms. We can cut it in half right to there, not all the way down, and it would have 2 heads. We can cut this sea star into 5 pieces and end up with 5 sea stars. So, you can't do that with like vertebrates, right? Take a fish, put it in a blender, it's done. It's not going to grow into more fish or anything like that. But a lot of these invertebrates can do that. And really, the sea star has a lot more in common with you than what you would think. And it can regenerate and grow into a bunch of different things. All right, so origin of multicellularity requires that basically new ways of cells to adhere and attach and signal and communicate with each other. Basically, they have to have the ability to communicate and send signals. And it's a little different than when we look at basically these choanocytes and then the collar cells as well. All right, so when we take a look at molecular analysis, so if you remember when we did the cladograms using the databases and we looked up the different animals online. So that's what molecular analysis has revealed similarities between genes coding for proteins. So basically we looked at the proteins, we can compare the proteins, and we can tell just by the proteins that their genes are very similar. There's very little base differences. Involved in adherence and attachment in choanoflagellates in animals. So we see some similar proteins there. And these choanoflagellates are the first ones to really have it, and then the animals continue with it. And so that's something that we see here only. This is the HOX gene that we see that's only found in these particular animals right here. And this has to do with connective tissue and basically holding the animals together. Early members of the animal fossil record include the Ediacaran biota, which dates back to about 560 million years ago. So that's basically 500 million years. Okay. Um, and so those are basically your earliest animal fossils. Early animal embryos and evidence of predation have also been found in the Neoproterozoic rocks. So basically, that's just a time in this date line where you can see these animals. So here's pictures of some of these animals, and you can see some animal characteristics. Here we can even see something that's advanced. See how we see how this repeating pattern here and here? That's a novel animal characteristic. Not all animals have that. If you think about a sponge, they don't have it. So, Cambrian explosion, about 535 to 525 million years ago, so again, about 500 million, marks the earliest fossil appearance of many groups of living animals. Basically, we have this, you have animals. They're really successful. There's lots of plants, vegetation, fungi, and protists on the planet. And now these animals come along and they're able to hunt and get their food. And so there's this explosion where we see all these new animals developing really, really rapidly because they're filling all these niches that, nothing was doing that before. Most of the fossils from the Cambrian explosion are of bilateral aliens. So animal development actually at first was a little slow, but then it starts to really, really, really speed up because of basically being bilateral. Bilateral symmetry begins to form. So think of a sponge, right? Sponges kind of asymmetrical, can't move around. We do have another type of symmetry called radial, and again, there's some advantages there to that. But realistically, we don't see as much in terms of the animals today, other than bilateral, because bilateral is where you have your true right and left half. And if you just think about like having a right and left half, that enables you to move forward, and you basically have a forward and a backward and a left and a right. And then we end up with a one-way digestive system. So these are all like novel traits that we're going to see develop. And this causes part of this Cambrian explosion because basically we have the basics for what are almost all of the animals today. All right, and so here we can see right and left half. There's a mouth. One would assume there's an anus somewhere back there. Mouth, anus, and yes, there is a right and a left half there on that one. And we look back here at these sponges, and no right and left half. As a matter of fact, they're kind of asymmetrical. These things are kind of radially symmetrical, and we'll talk about what that is here in a few moments. But you can even see some type of weird leg-looking structures there. So basically now you have new predator-prey relationships. You have things that are eating plants, you have things that are eating other things, and because of that, you're going to see a major change in the biota, in living things. A rise in atmospheric oxygen. Basically, you have all these photosynthesizers that have been ruling the planet for years, and now they're basically the oxygen levels are rising, and oxygen is really important for us in order to move around. So it's going to be the same thing with these animals. Now remember, a lot of this stuff's going to be ocean-based, so you got to have higher levels of oxygen in the air in order to have high levels of oxygen in the water as well. And then the evolution of the Hox gene, which is basically how we get more or less our repetition, our segmentation. So animal diversity continued to increase through the Paleozoic, and it was punctuated by mass extinction. So remember, we talk about punctuated equilibrium when we talk about evolution, where you have a lot of things develop, everything kind of steady for a little while, and then things die off. And then you get some new novel evolution, some new niches to fill, and then rapid evolution and a lot of things filling those niches. Vertebrates, which are things with a backbone, more or less, made the transition to land around 365 million years ago. And so we're going to see our first land vertebrates. So we're going to have coral reefs emerging. Ancestors of the plesiosaur were reptiles that returned to water. So do an image search real quick on a plesiosaur, and I'm betting you recognize that thing. It looks kind of like what a lot of people call the brontosaurus, except for it has little flipper things. It was not a dinosaur. And so during the Mesozoic era, dinosaurs were the dominant terrestrial vertebrates. So, we have animals move on to land. Notice this is halfway through the history of animals, and that's where we're starting to get these dinosaurs. And what makes a dinosaur is actually this pivot joint in their hip. This is why things like plesiosaurs and what are known as mosasaurs and pterodactyls are not considered dinosaurs. They're reptiles that fly. The pterodactyls are. The other ones are swimmers. All right. So, anyways, but yeah, they look a lot like dinosaurs. And a lot of times they're classified as dinosaurs by like in common terms. But really, when you look at scientific classification, no, they're not included. They're just really old animals that are now extinct. And then the first mammals are going to emerge right at the very end of this dinosaur reign. And mammals have some new abilities that the dinosaurs didn't bring to the table. But the issue is that they're not that much different than what the dinosaurs had, and therefore they weren't very successful because you have all these large predators just keeping the mammal population really, really low. And again, think of this idea of punctuated equilibrium. We have all these dinosaurs have a great advantage. They can move around really, really fast because of their hips. They expand, they take up a whole bunch of niches, and they go from 251 million years to 65.5 million years. So just doing some really rough math, that's about 200 million years. Notice that mammals are popping up around here really close to 65.5 million years. So mammals have, the dinosaurs, 200 million years, mammals less than 100 million years. And why didn't the mammal populations explode? Well, basically the dinosaurs get wiped out. They get wiped out by a meteor that just really changes their climate. There's some other issues as well, but really the meteors was the major tipping point. And this allowed the mammal numbers to increase, and now we end up with a lot more diversity of mammals shortly after this 65.5 million year extinction of the dinosaurs, kind of extinction. So first mammals are going to emerge. Flowering plants and insects diversified, and that's another contributing factor, is just the major changes in ecosystems. Dinosaur numbers were declining before the meteor hit. And so this opens the door for the mammals and allows the mammals to basically become more extensive. So beginning of the Cenozoic era followed mass extinctions of both terrestrial and marine animals, again because of that meteor thing. These extinctions include the large non-flying dinosaurs, pterodactyls, and the marine reptiles, your mosasaurs and plesiosaurs. And mammals increased in size and exploited the vacated ecological niches. So this is that punctuated equilibrium. We had something really disrupt life on Earth, and mammals had a lot of advantages that were similar to the ones that the dinosaurs had. A lot of good things going for them. They just couldn't increase in population and start to fill the niches that the dinosaurs did until the dinosaurs were out of the way. They're out of the way now, or moved on, and because of that, then there's a lot more room for the mammals to expand, grow, get much larger in size. Global climate cooled. This kind of tips an advantage towards the mammals a little bit, and because there's a lot of thought that a lot of the late dinosaurs were warm-blooded, so you can't argue too much on that, other than it did have an impact in mammals having hair were really well adapted. On the other hand, a lot of dinosaurs had feathers, so a lot of this information changes as we find out more about this extinction of the dinosaurs and then basically the rise of the mammals. So zoologists sometimes categorize animals according to body plan. So let's way back up here. Let's look at all these animals slowly. That was kind of a quick run through of animal evolution. So we're going to categorize them by body plan. Basically, your most basic animal is going to be your sea sponge. Sperm, fertilize the egg, blastula, that's it. And now you don't have body tissues. You just have a bunch of cells that can kind of differentiate, and that's your sea sponge. They are asymmetrical. That's your first major body plan. Some body plans have been conserved, while others have changed multiple times over evolution. And so another one of those ones is radial symmetry. They don't have a right and a left. Think of a jellyfish, and it's kind of organized like spokes, right? It's kind of got this circular arrangement. I mean, think maybe like a compact disc or a Blu-ray disc. All right, it's got this almost like tube shape to it. The disc is very flattened, but anything that's tube-like or circular-like, That's kind of the idea here with radial symmetry. There's no real front or back. If you think of a frisbee and you throw a frisbee, it really doesn't matter. It's going to go forward, but it's forward really as that disc is rotating. Any side can be forward, and with a jellyfish, that's very true. There's definitely an up and a down, but there's no front or back, and so food can come from any direction. It can move in any direction. If you think of like sea anemones, they just kind of sit there, and food can come from any direction, and it can sting it and capture it. So radial symmetry is kind of popular, and it's revisited in the animal kingdom. Basically, the radialins leave in one direction, and they just kind of stay that way. And bilateralians go another way, and we do revisit this radial symmetry kind of, sort of-ish. Not exactly when we look at like the sea stars. They're kind of radial, but really their larval stage is bilateral, and they're not perfectly radial. They're still really considered the bilateralians. So here we can see examples of your radial symmetry. Here's your sea anemone. Food can come from any direction. Whereas this bilateral symmetry, we have a right and a left half. Right and left half basically are closely similar. So two-sided symmetry is called bilateral. You have a dorsal and a ventral, so top and bottom, right and the left side, and then anterior and posterior. Now, anterior is usually defined as the end closest to the mouth. Sometimes it's the head because the mouth is found in a weird spot. And posterior refers to like the back end. It's the end, it's the closest to the tail. Now, not everything has a tail, it's closest to the anus. Again, usually there are some animals that have anuses in really weird places, or some animals don't even have an anus. So, that's kind of a difficult term sometimes, but generally speaking, when we think of like a quadruped, something with 4 limbs, or like even an insect which has 6 limbs, or like even centipedes which have hundreds of limbs, they have a head end, they have an anal end, and then they have all the legs and segments in between. Many also have sensory equipment such as brain concentrated in their anterior end. That's why we say the end closest to the head. Radial animals are often sessile or planktonic, and so we go back a couple pages, and here we go. This dude just waits for its food to come from any direction. That's why radial is an advantage to it. They eat small things. That's what planktonic means. Even jellyfish kind of drift about, and whatever they happen to bump into, they sting and then consume. Bilateral animals are more active. Basically, the right and the left allows you to be a better mover. Therefore, a better mover is more able to move more efficiently and get the food that it needs more efficiently. And they have a central nervous system versus a nerve net, which we see in a lot of the radiolarians. So, animal body plans also vary according to the organization of the animal's tissues. So, basically, tissues are collections of specialized cells. We don't really truly have this in sea sponges. I mean, they kind of have differentiated types of cells, but are they really true tissues? Not so much. And again, this is why a lot of scientists want to kind of kick the sponges out of the animal phyla. And so when we refer to true animals, we call them eumetazoans. And that basically includes everything but sea sponges. So during development, you end up with 3 germ layers, and that give rise to the tissues and the organs of the animal embryo. And actually, early on, we don't even see 3 germ layers. Really, we just see 2. We have an ectoderm, which is the outside. So what's going to come out, form from the outside? We mentioned this already. What's on the outside of all animals? Some type of skin. That's your ectoderm. Now, another tissue that comes from that is the nervous system, because we end up a little bit of this tissue folds in and becomes the nerve cells. The endoderm is the innermost, and that basically is going to become your digestive tube. And so animals that have 2 layers are going to be diploblastic. Sponges and a few other groups lack true tissues. Diploblastic animals have an ectoderm and an endoderm. These include the cnidarians and a few other groups. Cnidarians are your jellyfish and your sea anemones. So they fold in, they form a blastula, they form a gastrula, and then they get off the animal train. That's as far as they go developing as animals go. And these guys are all considered the radiolarians. In other words, they have radial symmetry. Then we see the triploblastic, and triploblastic basically goes hand in hand with being bilateral. And these animals have a mesoderm, a middle layer. So what's found between your skin and your digestive system? Muscles, connective tissue like bones, cartilage, things like that. Your kidneys are also part of that mesoderm. So these include flatworms, arthropods, vertebrates, and other animals. So flatworms, your parasitic worms, basically, is what you would normally think of. You have your arthropods, which are like insects, crabs, crustaceans, things that are crunchy on the outside, and then vertebrates, things with backbones. Most triploblastic animals possess a body cavity. A true body cavity is called a coelom and is derived from the mesoderm. So we have this third layer develop. It's a middle layer, and that middle layer is going to line a space. Where do you store your lungs, your heart? Is that a different place than your stomach, your intestines, your liver? All right, all those things are in this inner body cavity. If there isn't an inner body cavity or coelom, you aren't going to be able to store them. Now, your coelom is divided further into more cavities. But coelomates are animals that possess a true coelom, a true body cavity. So we look here. Here's an acoelomate. Notice there's no body cavity. Really, you got a digestive system, you got a skin, and you got some muscular tissue in between. There's no space for holding things like lungs. So you aren't going to have those things. Now, here we have a body cavity, but it's only lined on the outside, so these guys are called pseudocoelomates. Pseudo means fake, and these are basically your roundworms. They have some advancements, but again, not all the complex organs. Then we look at an earthworm, and an earthworm has everything, basically every system that we have. Skeletal system is kind of an exception there, but other than that, they've got like all the major systems that we have. And so therefore, that's why they have this coelom or body cavity for storing all of those organs. So most animals are coelomates because, again, having organs, complex organs, is a big advantage. Here's your pseudocoelomates, your acoelomates. All right, so pseudocoelom is a cavity derived from mesoderm and endoderm. Triploblastic, again, have three body layers, endoderm, ectoderm, and mesoderm. Be familiar with what those different tissues, what they turn into. Triploblastic animals that lack a body cavity are called acoelomates, so those are our flatworms. Again, just kind of repetitive of what we've been talking about the last couple of pages. A grade is a group whose members share key biological features. So if you think about it like coelomates is a grade, there's a lot of animals that have body cavities. Like earthworms have body cavities. I really don't think I'm that closely related to an earthworm. So that's why that's considered a grade. A grade is not necessarily a clade, an ancestor of all of its descendants. And so remember, we do collectively call them this generic term of coelomates, but that doesn't mean they're all that closely related. They've diversified a lot. It's a key trait that just kind of kept on going. Now, the last major thing that we divide animals into is whether they're a protostome and deuterostome. And you actually already know what these are if you've been paying attention. Based on early development, many animals can be categorized into having a protostome development or deuterostome development. What the heck is this? Well, remember what I told you about early on, that cleavage and whether it is rotated or lined up very well. So in protostome development, cleavage is spiral and determinate. In other words, when you have that morula, you have that 8-stage cell, you can tell either they're lined up or they're not lined up. If they're not lined up, then we call that spiral, and then that's called determinate. In other words, those cells have determined basically some of their fate along the future. They've already determined whether they're a head or a tail end. So that's in protostome development. In deuterostome development, cleavage is radial and indeterminate. And I'm thinking I said something wrong here a second ago about protostomes and deuterostomes, but so let's just review this real quick. So here we have protostomes, cleavage is spiral, determinate. In other words, it's rotated a bit. It's the fate's already been determined. Whereas in deuterostomes, the cleavage is radial and it's indeterminate. In other words, any of these cells can become another cell. So let's take a look here. Here we go. So here we have spiral and determinate. If you remove any of these cells, it's already determined that it's going to be the head or the tail. These ones we can remove and they can grow into a whole nother basically bundle of cells. And that's how you end up with identical twins. So in other words, you can't end up with identical twins here in these protostomes. So protostomes are going to be most of your invertebrates. The blastopore forms first. That's going to end up being the mouth. And some of these guys don't have anuses, but eventually this tube will become a tube and will become an anus. This is the digestive tract. Over here, this is radial. The cells are lined up. It's indeterminate. The whole cell mass can divide and become identical twins. And here, this blastopore more or less becomes the anus. Notice that we have a difference in how the mesoderm, or this red layer of tissue, develops too. These cells just kind of flake off and grow in the middle, whereas these ones actually fold in again, and then they're going to fill up this area right here. So here we have the anus and the mouth. Here we have the mouth and then the anus. So we go back. So indeterminate cleavage makes it possible for identical twins, embryonic stem cells, yada yada. Again, we only see that in the vertebrate lineage, and then there's one other group that goes along that lines. So more or less, this separates vertebrates from invertebrates. Here we have our mollusks like squids and clams. You have your annelids like earthworms. Insects are going to belong to this group, your arthropods. Whereas over here we have the chordates, which basically means, more or less, it means you have a backbone. But it also includes the echinoderms. Echinoderms, I've also pronounced echinoderms, are going to be your sea stars. So that's just an odd group that we have that's connected with us. Now, if you did dissection with me, when we dissected the sea star, you saw this skeleton or this bony plates on the inside. So notice that the ones that have endoskeletons are over here, and the ones that have exoskeletons are over here. So another difference between protostomes and deuterostomes. All right, so again, just some comparisons over the last couple of pages. Protostome development, the splitting of solid masses of mesoderm forms a coelom. In deuterostome development, the mesoderm buds from the wall of the archenteron to form the coelom. All right, what does this mean? So go back here and right here. These cells just flake off and become the mesoderm in protostomes. In deuterostomes, it buds or folds in, and then we get these red cells taking up this space and this space right there. The blastopore forms during gastrulation and connects to archenteron to the exterior of the gastrula. In protostomes, that blastopore becomes a mouth. In deuterostomes, the blastopore becomes the anus. And again, we started the lecture off with that discussion. By 500 million years ago, the animal phyla with members alive today were established. So animals, once they move on to the land, we have all the phyla of animals for the most part by 500 million years ago. And so, animal development was relatively rapid, and we end up with these phyla relatively rapidly, and then it's just kind of been steady since then. And you might say, well, what about the development of dinosaurs? What about the development of amphibians and fish? Like, all those things happened more recently, much more recently than 500 million years ago. Well, it says all of the phyla. You got to remember that all things with a backbone belong to the same phylum. So the chordates, which are things, again, more or less with a backbone, just like the real loose, simple definition, they were established even, around this 500 million years ago time frame. So zoologists recognize about 3 dozen animal phyla. Phylogenies now combine morphological, molecular, and fossil data. And again, back when we were looking at cladograms, we talked about how it's important to look at not just body shape and structure, but also the molecular data and the fossil data. So now we have a much better understanding of the animal relationships. So, 5 important points. All animals share a common ancestor. Sponges are basal animals. In other words, they are just barely animals. They hardly have any of the characteristics, but they have the basic characteristics of what makes an animal. Multicellular, eat other things, sexual reproduction. That's about it. Eumetazoa are your true animals, and they're the animals with tissues. So basically, every other animal fits into that category. Most animal phyla belong to the clade Bilateralia. And so we have the radials and the bilaterals, and they're kind of we diverge into these two major groups, but almost the most successful one are the bilaterals, and they kind of take over. There are 3 major clades of bilateral animals, all of which are invertebrates. The animals that lack a backbone except for chordata. Chordata are your vertebrates, and that's because they have a backbone. So you have a metazoan, that basically means to be an animal. Sponges, basically everything fits into that, but sponges, that's as much animal as they can get. Eumetazoa are your true animals, and we're going to have multiple tissue layers. And here we have some of these radiolarans. The bilaterals are now everything else. That's an advantage. Now it's such a good advantage that all the rest of the animals have that. That even includes a lot of these worms. Then we have our deuterostomes, and now we start developing the true anus and such. And that's a major advantage, like having a complete digestive system. So that's all of these guys. And then you have your protostomes. I did this backwards. All these guys are protostomes. And then here are our deuterostomes, and that includes your hemichordata, your echinochordata, and your chordates. So here's your deuterostomes. And these guys, so all of these guys have complete digestive systems. Here are your deuterostomes. These are the guys that now have your most advanced type of embryonic development, where the anus actually develops first, the mouth develops later, and not only do they have complete digestive systems, basically a more advanced development. Okay, so, and again, these are the protostomes down here, and these guys are the first ones to have. See, these nematodes were the first lowest group to have a complete digestive system. So again, just a closer look at how all these things, the different characteristics that we see that are important and how long ago they developed. Notice that within 100 million years, which is less time than the dinosaurs ruled the Earth, we went from just barely being an animal to having that bilateral symmetry. And the bilateralians, okay, so here are your protostomes. We have 2 major groups here. We're not going to get into what the differences are there, but mouth develops first. We have a complete digestive system, and these ones not up here. And then we have segmentation and such. We end up with different limbs. And up here we have the deuterostomes. Anus develops first, mouth later, endoskeleton, so on and so forth. All right, so bilateralians are divided into 3 clades: deuterostomes, ecdysozoa, and lophotrochozoa. Deuterostoma includes hemichordates, which are called acorn worms. I'm not going to spend any time really talking about those guys. Echinoderms, which are sea stars, sea urchins, those relatives, and then the chordates. The clade includes both vertebrates and invertebrates for deuterostomia. Ecdysozoa is a clade of invertebrates that shed their exoskeletons. So basically you have an outer crunchy shell. Lophotrochozoa is another clade of bilateral invertebrates. Basically it's all the other ones. They have a larval feeding stage, feeding structure called a lophophore. and then they have a larval stage called a trochophore. So some other questions they have are sponges monophyletic? There's some major groupings and differences in the development of sponges. There's a lot of questions about whether they're really even animals. Um, Ctenophora is another group of animals. It's kind of an outlier. So if we kick sponges out of the animal phyla and say they're just another type of protist, then our ctenophores really are basal metazoans. Our acoelomate flatworms, basal bilateralians, basically they just barely have a right and a left half. A lot of times you can't even tell with roundworms what their right and left half is unless you kind of rotate them around a little bit and look. So again, just some future questions and future research to be answered. We're not going to do anything with this chart. And again, just some breakdown of when these major traits appeared as we look at it on a timeline. Again, just kind of a review of the traits we've been talking, another way of looking at it. You end up with your true tissues, so there's your eumetazoan, bilateral and 3 layers, those are your bilateralians, and those are most of your animals. So another chart breaking it down, take your time to look at that, but I'm going to go ahead and just move on. Just kind of repetition of what we've been covering before. Okay, anyways, that's it for your basics on animals.